All right, guys, last chapter for Unit 3, okay? So we are going to do a test for this next week. And like I said, I'll do a review session live. I'm looking pretty good. Talk to Kite, and we're going to do a practice live before we do the review session live, okay? And then we'll have our test. So, chapter 39, plant responses to internal and external signals, all right? Here we go. Stationary life. Plants can't move, right? They get eaten, they can't move. So they have to have some other ways of handling things. So plants respond differently to stimuli than animals because they're stationary. Plants have to change their patterns of growth and development. We've all done the little experiment or seen the little experiment done, right? If you pick a plant and you put it in a little labyrinth, you know, is it going to grow towards the light? And of course the answer is always yes. The plant's going to grow towards that light. That's what they mean, okay? The consequence is a greater difference in their body forms than is seen in animals, right? Because you're not going to see an animal that's leaning this way because it had to grow towards the light. A giraffe is always going to be standing up straight. It'll just move to go to its food. The food source, sunlight, for plants, is going to cause the tree to curve, depending on where it needs to go to get that sunlight in order to be able to produce its own food. That's what they're talking about here. So we're talking about signal transduction pathway. For those of you who were in my day class, you're going to remember this part. For those of you who were in my night class, I don't believe we went over this, so it's okay. It's not a big deal. There's 3 parts to receiving a signal. Actually, even in my night class we went over it because we went over it for the final exam. There's 3 steps to receiving a signal in the body, whether it's a human body or the plant, all right? Reception of the signal, transduction, response. I believe I told you guys the football analogy, right? You're going to receive the football, you're going to run the football, and when you make the score, you're going to do your little football dance, right? End zone dance. I'm not doing the end zone dance any longer because this is being recorded. Reception. Reception, protein receptor, right? You've got the protein receptor right here. It's going to receive that signaling molecule. That signaling molecule can be a hormone, it can be environmental stimulus, it can be anything, okay? The transduction, the relay molecules, the processing of that receptor molecule or that stimulus. So it's processing it, it's understanding what's going on, and then whatever necessary cellular response is going to occur. That's what's happening. That's the 3 steps, okay? Reception, transduction, response. Reception, transduction, response. Know those 3 steps. Pretty please. Reception signals are first detected by the receptors. Proteins are going to change their shape. Nothing new here. Transduction. Each chemical in the transduction pathway is capable of initiating multiple next steps. So a lot of times these are multiple steps that go along the way. So right here, they're showing you, right? Here it's a reception of light. Oh, wait, I get a light. I've got a second messenger is produced. I'm going to have to go ahead and have protein kinase 1 activated, which is going to cause the actual transcription to start. Remember, transcription going from DNA to RNA, right? Then here, you've got calcium. All of a sudden, calcium is getting pumped into the cell. Oh, look at that. We're going in, all right? That's going to have kinase 2 activated. That's going to cause a second transcription to occur, okay? That's going to cause transcription to leave. Translation is going to occur. Translation, messenger RNA to your protein, or before that, amino acid, which eventually leaves your protein, and then you've got your proper response. All right? I know we're reviewing a lot of information at once, so let me slow down for a minute. So transcription, DNA to messenger RNA, DNA, A, T, G, C, messenger RNA, A goes to, please tell me you didn't say T, U, T goes to A, G, C, C, G, right? And then your messenger RNA is going to go to your amino acid. We're going to read this 3 letters at a time because that's your codon, right? And I don't have the codon chart up, but I'm going to pull it up in just a heartbeat. UAC, can read it with either one of these. UAC, UAC, right? 9 o'clock, 12 o'clock, 3 o'clock. That's tyrosine, or using this circular chart, UAC, tyrosine. Let me go back to my PowerPoint. That's your tyrosine, right? And this is translation. Just a little review, right? No, you won't get tests on this stuff. I just want to make sure you remember this, right? Good old times. Okay. And then finally, your response. Responses can be changes in the regulation of one or more cellular activities. You have two main mechanisms. Stimulate the transcription of messenger RNA for the enzyme, transcription regulation. Or you can activate the existing enzyme molecules that are in the inactive state. So something that was in a pause situation gets reactivated, turned back on. Post-translational modification, you turn it back on. All right? Okay, plant hormones. Believe it or not, plants have hormones. Hormones are substances that will bind to specific receptors and triggers responses in cells and tissue. You don't need a lot, just like with us. Excuse me, you don't need a lot. Small amounts are needed to trigger large responses. And depending on what the response you want, you're going to have different hormones. There's a lot of hormones in here. There's only specific ones I want you to know about. Auxin. Auxin is a hormone that's actually going to stimulate the elongation of cells and the developing of shoots. So what time of year would you expect to see a lot of auxin? Right now, right? Because it's springtime. Our shoots are growing, so you would expect to see high levels of auxins. So in theory, could you test your plant to see if it has auxins, if it's perhaps not growing, to see if there's something wrong with it? You could. Do they actually do this? I have no idea, quite honestly. But maybe they do. Oh, heads up, I've had coffee, good luck. Coffee's kicking in, I can feel it. Cytokinins. Hopefully we remember cytokinins from the cell cycle. Remember our 3 pauses in the cell cycle? Let's go through the cell cycle real quick. Remember the phases? G1. If we have a G1, we have to have a G2. And it's not going to be right after it, because if it was right after it, then they would have just put them together, right? So it's going to be a cross, G2. Okay. Then we have the S phase, S for synthesis, for making a copy of DNA. And then, of course, you have the M phase, which is mitosis and cytokinesis together, right? So you had a checkpoint here, you had a checkpoint here, and you had a checkpoint here, right? 3 different checkpoints to make sure that the cell really needs to go to the next part of its cell cycle. The actual molecules that were in charge of those checkpoints were cytokinins. So just like in animal cells, plant cells also have cytokinins, and they're also involved with cell division and influence the pathway of cell differentiation. Remember, cell differentiation is what is going to make a cell be, for plants, either part of your dermal tissue, ground tissue, or vascular tissue, right? So cytokinins are very, very important. Eumerillins, they'll function in stem elongation, fruit growth, and seed germination. So it's going to be really important, especially in the later springtime and summer as well. Bacinosteroids, similar in structure to steroids in humans, found in seeds, fruits, shoots, leaves, and floral buds. They'll inhibit root growth, delay the falling of leaves, and promote the differentiation of xylem cells. Remember, xylem carries water, right? WX, follow the alphabet. That's for your water. Abscisic acid, APA. Originating thoughts have an important role in leaf abscission and bud dormancy, so this is what we're going to see more towards the fall, late fall, early winter. It causes drought tolerance by forcing the closure of the stomata for the opening of the potassium channels, so this was obviously also be very important in desert plants. Six more slides, guys, 6 more slides. Strigolactones, it's going to promote seed germination and controls the apical dormancy. So again, it's going to be, apical dormancy is going to be in right before winter, and then seed germination is going to be right around springtime. All right. You got the hormone at the very top, and then ethylene. Okay, I love ethylene. Our bananas. What is the worst thing you can do with your bananas? Put them right next to all of your fruits and vegetables, right? Because bananas are extremely high in ethylene. If you have a chance to go to the grocery store and look at your bananas, you might notice a lot of the bananas actually have a little saran wrap on the stems of them. And that's because in the stem, you have a lot of ethylene. And when you don't protect, when you don't cover up that, the ethylene just it gets released like crazy, and any fruits or vegetables nearby are going to ripen very fast. Now, on a good note, if you have fruits that you've bought that are not quite ripe yet, if you put them next to bananas, oh my gosh, in a day or even 2 at the most, they'll be completely ripe, okay? Because of the amount of ethylene found in bananas. It's really kind of interesting. So it's very important for fruit ripening. Light response. Light is the trigger for many major events in plant growth and development. Well, no, that's not a surprise at all, considering how important it is for photosynthesis. I think that's a good one, yep. Biological clocks and circadian rhythm. Circadian rhythm can change in various processes depending on the time of day, which are not controlled by any known environmental factors, right? So that's why whenever we go through daylight changing time, it always takes us a while, a good week, sometimes even 2, to get caught back up into it. We need anything that messes up our pattern, we don't do well. That's why it's a gradual change, so that 1 hour time difference messes us up. Without access to sunlight, biological clocks do not run on a 24-hour day. It's just, plants don't do well. They like that gradual change. We know December 21st, roughly, that's the shortest day of the year, and slowly we go back up, we get more time. Plants deal well with that gradual increase. June 21st, roughly, longest day of the year, they do great, and then they slowly decrease. Well, 2 more slides. Photoperiodism. Photoperiods are relative lengths of the day versus night. These change with the seasons. We know this. Short day plants will only flower when the amount of sunlight is less than a critical length. Long day plants will only flower when the amount of sunlight is more than a critical length. So that's why you've got certain plants that'll tell you when you buy plants, it'll say these will flower in summertime, these will flower in autumn. That's what we're talking about right here, okay? Day neutral plants flower based on cues other than light, such as maturity or water levels, actually controlled by the amount of dark rather than the amount of light. There's some plants that will only flower at nighttime. Night blooming jasmine would be one of them. Red light interrupts nighttime periods the best. So what kind of defenses can plants have against herbivores? Plants use defenses to counteract herbivory. We've talked about this, prevent infection. Some of them are thorns, having a tougher bark, thick cuticle. Some of the chemical defenses they have, we've talked about this, cinnamon, right? Having a distasteful taste or a toxic compound, poisonous. Release of volatile chemicals to prime nearby plants and recruit insect helpers. We've talked about those examples. All right, guys, we are done. We have completely finished plants.